Evidence of evolution. By the end of this video, you will be able to explain sources of evidence for evolution and define homologous and vestigial structures. Welcome back to Concepts of Biology. We'll continue to read section 11.3. The evidence for evolution is compelling and extensive. Looking at every level of organization in living systems, biologists see the signature of past and present evolution. Darwin dedicated a large portion of his book on the origin of species, identifying patterns in nature that were consistent with evolution, and since Darwin, our understanding has become clearer and broader. Fossils. Fossils provide solid evidence that organisms from the past are not the same as those found today. Fossils show a progression of evolution. Scientists determine the age of fossils and categorize them all over the world to determine when the organisms lived relative to each other. The resulting fossil record tells the story of the past and shows the evolution of form over millions of years. For example, highly detailed fossil records have been recovered for sequences of species in the evolution of whales and modern horses. The fossil record of horses in North America is especially rich, and many contain transition fossils, those showing intermediary anatomy between earlier and later forms. The fossil record extends back to a dog-like ancestor some 55 million years ago that gave rise to the first horse-like species 55 to 42 million years ago in the genus Eohippus. The series of fossils tracks the change in anatomy resulting from a gradual drying trend that changed the landscape from a forested one to a prairie. Successive fossils show the evolution of teeth shapes and foot and leg anatomy to a grazing habit, with adaptations for escaping predators, for example in the species of Mesohippus found from 40 to 30 million years ago. Later species showed gains in size, such as those of Hipparion, which existed from about 23 to 2 million years ago. The fossil record shows several adaptive radiations in the horse lineage, which is now much reduced to only one genus, Equus, with several species. Figure 11.10. This illustration shows an artist's rendering of these species derived from fossils of the evolutionary history of the horse and its ancestors. The species depicted are only 4 from a very diverse lineage that contains many branches, dead ends, and adaptive radiations. One of the trends depicted here is the evolutionary tracking of a dry climate and increase in prairie versus forest habitat reflected in forms that are more adapted to grazing and predator escape through running. Przewalski's horse is one of a few living species of horse. Anatomy and embryology. Another type of evidence of evolution is the presence of structures in organisms that share the same basic form. For example, the bones in the appendages of a human, dog, bird, and whale all share the same overall construction. That similarity results from their origin in the appendages of a common ancestor. Over time, evolution led to changes in the shapes and sizes of these bones in different species, but they have maintained the same overall layout, evidence of descent from a common ancestor. Scientists call these synonymous parts homologous structures. Some structures exist in organisms that have no apparent function at all and appear to have residual parts from a past ancestor. For example, some snakes have pelvic bones despite having no legs because they descended from reptiles that did have legs. These unused structures without function are called vestigial structures. Other examples of vestigial structures are wings in flightless birds, which may have other functions, leaves on some cacti, traces of pelvic bones in whales, and the sightless eyes of cave animals. Figure 11.11. The similar construction of these appendages indicates that these organisms share a common ancestor. Another evidence of evolution is the convergence of form in organisms that share similar environments. For example, species of unrelated animals, such as the Arctic fox and ptarmigan, which is a bird, living in the Arctic region have temporary white coverings during winter to blend with the snow and ice. The similarity occurs not because of common ancestry, Indeed, one covering is of fur and the other of feathers, but because of similar selection pressures, the benefits of not being seen by predators. Figure 11.12. The white winter coat of the Arctic fox and the ptarmigan's plumage are adaptations to their environments. Hi! If you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. Alright, let's get back to reading. Embryology, the study of the development of the anatomy of an organism to its adult form, also provides evidence of relatedness between now widely divergent groups of organisms. Structures that are absent in some groups often appear in their embryonic forms and disappear by the time the adult or juvenile form is reached. For example, all vertebrate embryos, including humans, exhibit gill slits at some point in their early development. These disappear in the adults of terrestrial groups, but are maintained in adult forms of aquatic groups such as fish and some amphibians. Great ape embryos, including humans, have a tail structure during their development that is lost by the time of birth. The reason embryos of unrelated species are often similar is that mutational changes that affect the organism during embryonic development can cause amplified differences in the adult, even while the embryonic similarities are preserved. Biogeography. The geographic distribution of organisms on the planet follows patterns that are best explained by evolution in conjunction with the movement of tectonic plates over geological time. Broad groups that evolved before the breakup of the supercontinent Pangaea, about 200 million years ago, are distributed worldwide. Groups that evolved since the breakup appear uniquely in regions of the planet, for example, the unique flora and fauna of northern continents that formed from the supercontinent Eurasia and of the southern continents that formed from the supercontinent Gondwana. The presence of Proteaceae in Australia, Southern Africa, and South America is best explained by the plant family's presence there prior to the southern supercontinent Gondwana breaking up. Figure 11.13. The Proteaceae family of plants evolved before the supercontinent Gondwana broke up. Today, members of this plant family are found throughout the southern hemisphere, shown in red. The great diversification of the marsupials in Australia and the absence of other mammals reflects that island continent's long isolation. Australia has an abundance of endemic species found nowhere else, which is typical of islands whose isolation by expanses of water prevents migration of species to other regions. Over time, these species diverge evolutionarily into new species that look very different from their ancestors that may exist on the mainland. The marsupials of Australia, the finches of the Galapagos, and many species on the Hawaiian Islands are all found nowhere else but on their island, yet display distant relationships to ancestral species on mainlands. Molecular biology. Like anatomical structures, the structures of the molecules of life reflect descent with modification. Evidence of a common ancestor for all of life is reflected in the universality of DNA as the genetic material and of the near universality of the genetic code and the machinery of DNA replication and expression. Fundamental divisions in life between the three domains are reflected in major structural differences in otherwise conservative structures such as the components of ribosomes and the structures of membranes. In general, the relatedness of groups of organisms is reflected in the similarity of their DNA sequences, meaning exactly the pattern that would be expected from descent and diversification from a common ancestor. DNA sequences have also shed light on some of the mechanisms of evolution. For example, it is clear that the evolution of new functions for proteins commonly occurs after gene duplication events. These duplications are a kind of mutation in which an entire gene is added as an extra copy or many copies in the genome. These duplications allow the free modification of one copy by mutation, selection, and drift, while the second copy continues to produce a functional protein. This allows the original function for the protein to be kept, while evolutionary forces tweak the copy until it functions in a new way. In our next video, we'll read section 11.4, Speciation.